Hello, I'm Dr. William Schlosser, Washington State University, School of the Environment. This is my classroom.
The study of ecology considers population growth with birth rates and death rates, regulation and intraspecific and interspecific competition, mutualism, and predation. Focusing on adaptations, physiological ecology is concerned with the responses of individual organisms to temperature, moisture, light, and other environmental conditions. Closely associated with population and evolutionary ecology is community ecology, which deals with the physical and biological structure of communities and community development. To launch into inquiries, we investigate population genetics. Population ecology is evolutionary ecology that deals with the role of natural selection in physical and behavioral adaptations and speciation. Look at this image to identify populations, but you will also identify the biome they exist in and how these environmental differences lead to speciation. Is a Douglas fir tree in Yellowstone National Park genetically identical to the Douglas fir in the Olympic Mountains of Washington? How about elk or the wolves in each location? Are they all identical genome species? We will consider the ideas of generalists and specialist species existing in different ecosystems. The process in which one species gives rise to multiple species that exploit different features of the environment, such as food resources or habitats, is called adaptive radiation. The different features of the environment exert the selection pressures that push the population in various directions, and reproductive isolation, the necessary condition for speciation to occur, is often a byproduct of the changes in morphology, behavior, or habitat preferences that are the actual targets of selection. Speciation is the evolutionary process by which populations evolve to become distinct species. Charles Darwin was the first to describe the role of natural selection in speciation in his 1859 book, The Origin of the Species. He also identified sexual selection as a likely but problematic mechanism. There are 4 geographic modes of speciation in nature, based on the extent to which speciating populations are isolated from one another: Allopatric, Peripatric, Parapatric, and Sympatric. Focus on these terms, know how to define them, apply them to populations you observe, and be able to give examples of how you understand these instances. Speciation occurs when biological populations of the same species become isolated from each other to an extent that prevents or interferes with gene flow. The separated populations develop adaptive responses to their restrictive environments. We look for the examples to prove it. We are looking at racks of 2 5-point bull elk I harvested, and their body sizes were very similar. The bleached one on the right was left in the sun too long. It was colored like the one on the left when I shot it. The rack on the left was kept under closed storage until I mounted it. That smile in the middle is my daughter. She is not an elk. I harvested the Rocky Mountain elk, Cervus canadensis variety nelsoni, shown on the right in 1991, at Bald Mountain, Selway River Drainage near the Nez Perce Trail at Bald Mountain, Idaho. The Manchurian wapiti, Cervus canadensis variety zachopygus, was shot in Khabarovsky Krai, Russian Far East in 1996. These are found to the east of Lake Baikal, Russia. As populations, they have common ancestry, but have been genetically separated for 14,500 years. I take you back several lectures to remember the geologic conditions confirming the current ice age we are still exiting from. Only about 21,000 years ago, the current ice age was at its crescendo. Surface waters were mostly frozen, locked in terrestrial glaciers and sea ice. This meant there was less water resting in the oceans. Sea levels stood about 120 meters, 400 feet lower than it does today. That means the Bering Straits were open fields and boreal forests, connecting these two continents. Thuringia is a land in the maritime area bounded on the west by the Lena River in Russia, and on the east by the Mackenzie River in Canada, on the north by 72 degrees north latitude in the Chukchi Sea, and on the south by the tip of the Kamchatka Peninsula. This territory was an easily traversed path between these two continents. Species of all types were connected along this connection, linking current-day Alaska with the Russian Far East. The connective tissue now under the North Sea was about the size of today's country of Mexico. At that time, 21,000 years ago, the world was in the apex of the Ice Age, and the elk species were connected as a series of populations along this continental connection zone. Genetic materials were still shared between populations. Both six-point bulls in these photos are about the same structure and build. Genetically, these two subspecies have been separated by 8,550 kilometers, or 5,500 miles, and about 14,500 years. Restrictive environments faced by each population favored random mutations to solve similar adaptation needs. They may look similar, but the DNA of these representatives of historically related species is no longer identical. It is not similar enough to support genetically viable offspring made between mates of these two populations. Remember, viable offspring require production of offspring who can also successfully reproduce. Just because offspring are made, it does not mean that they can also reproduce. Think about mules for this hybrid situation. In this example, a geographic change separated members of the elk populations into more than one group. Such changes could include the formation of new mountain range or a new waterway, or the development of new canyons. Here we see how lush boreal forest ecosystems were flooded. It was a warming, and that connected the Arctic Ocean to the Pacific Ocean. Different gene mutations in each subpopulation occurred and built different solutions to similar restrictive environments. The different mutations and resultant genetic variations led to different characteristics between the separated populations. Allopatric speciation has occurred. Allopatric speciation is based on the extent to which associated populations are isolated from one another. The 400-foot deep Thuringia Ocean is a significant barrier for elk to pass, but geographic barriers appear in different formations and create temporal discontinuities. Now we consider Peripatric Speciation. Parapatric Speciation is a mode of speciation in which a new species is formed from an isolated peripheral population. Since Peripatric Speciation resembles Allopatric Speciation, in that populations are isolated and prevented from exchanging genes, it can often be difficult to distinguish between them. Nevertheless, the primary characteristic of Parapatric speciation proposes that one population is much smaller than the other. The term peripatric is often used in biogeography, referring to organisms whose ranges are adjacent but do not overlap, being separated where these organisms do not occur. For example, on an oceanic island compared to the mainland. Such organisms are usually closely related and sometimes called sister species. Parapatric can be distinguished from allopatric speciation by three key features. 1. The size of the isolated population. 2. Strong selection caused by the dispersal and colonization of novel environments. 3. The effects of genetic drift on small populations. The size of a population is important because individuals colonizing a new habitat likely contain only a small sample of the genetic variation of the original population. This promotes divergence due to strong selective pressures, leading to the rapid fixation of an allele within the descendant population. This gives rise to the potential for genetic incompatibilities to evolve. These incompatibilities cause reproductive isolation, giving rise to sometimes rapid speciation events. Red spruce, Picea rubens, has arisen from an isolated population of black spruce, Picea mariana. During the Pleistocene, a population of black spruce became geographically isolated, likely due to glaciation. The geologic range of the black spruce is much larger than the red spruce. Red spruce has significantly lower genetic diversity in both its DNA and its mitochondrial DNA than the black spruce. Furthermore, the genetic variation of the red spruce has no unique mitochondrial haplotypes, only subsets of those in the black spruce, suggesting that the red spruce speciated peripatrically from the black spruce population. It is thought that the entire genus Picea in North America has diversified by the process of Peripatric Speciation, as numerous pairs of closely related species in the genus have smaller southern population ranges, and those with overlapping ranges often exhibit weak reproductive isolation. In these first two examples, we saw the forceful abiotic powers of the current ice age to alter environments and create geographic barriers to how species share genes through time. Now we look at a spin to this with the name Parapatric Speciation. In Parapatric Speciation, one population bifurcates into two subpopulations of a species, evolving in reproductive isolation from one another, while continuing to exchange genes. This mode of speciation has three distinguishing characteristics. 1. Mating occurs non-randomly. 2. Gene flow occurs unequally. And 3. Populations exist in either continuous or discontinuous geographic ranges. This distribution pattern may be the result of unequal dispersal, incomplete geographic barriers, or divergent expressions of behavior, among other things. Parapatric speciation predicts that hybrid zones will often exist at the junction between the two new populations. Natural selection is the primary driver in parapatric speciation, and the strength of the selection during divergence is often an important factor. Parapatric speciation may also result from the reproductive isolation caused by social selection, that is, individuals interacting altruistically. Due to the continuous nature of parapatric population distribution, population niches will often overlap, producing a continuum in the species' ecological role across an environmental gradient. Reduced gene flow of parapatric speciation often produces a cline in which a variation in evolutionary pressures causes a change to occur in allele frequencies within a gene pool of each differentiating population. This is enabled through environmental gradients that ultimately results in genetically distinct sister species. This is parapatric speciation. Sympatric speciation is the evolution of a new species from a surviving ancestral species, while both continue to inhabit overlapping geographic regions. In evolutionary biology and biogeography, sympatric and sympatry are terms referring to organisms whose ranges overlap so that they occur together at least in some places. This is the case with deer species in North America. White-tailed deer, Odicoleus virginianus, is the native deer species in this continent. Through time and geographic habitat differences, sympatric speciation has given rise to mule deer, Odicoleus hemionus, to fill niche opportunities in higher elevation mountainous regions, steeper river basin habitats, and specialized feeding characteristics common to these lands. In some western regions of North America, the white-tailed deer, Odicoleus virginianus, range overlaps with those of the mule deer, Odicoleus hemionus. Kemiak Butte supports populations of both species. They intermingle during the same periods of the year and on the same sites. Here, these species representatives appeared on wildlife cameras on the south-facing aspect within a couple of weeks of each other. White-tailed incursions in the Columbia River Basin in southeast Washington and north-central Idaho have witnessed some hybridization evidence. I've witnessed white-tailed deer does about 15 miles southwest of the WSU campus near Almota, Washington, with hybrid fawns. These offspring demonstrated difficulty running like a white-tailed deer, but could not hop like the mule deer genetic contributors either. In a rush, these fawns fell miserably. Evidence suggests that most male offspring are lost in vitro. Surviving males are sterile. Female offspring often live to maturity, but most of these are infertile. Further evidence suggests that fertile does can only breed with 100% pure males of either species, thus making offspring that are 75% one species and 25% the other. Evidence has not been generated to confirm if embryo males of those unions can survive to maturity, or if they are reproductively fertile. In North America, the white-tailed deer species are widely distributed east of the Rocky Mountains, as well as in southwestern Arizona and most of Mexico, aside from Lower California. It is mostly replaced by the black-tailed deer or mule deer from that point west, except in mixed deciduous riparian corridors, river valley bottomlands, and lower foothills in the northern Rocky Mountain region from South Dakota west to eastern Washington and eastern Oregon, and north to northeastern British Columbia and southern Yukon, including in the Montana Valley and the foothill grasslands. White-tailed deer are not found in western Washington, existing only in the eastern half. The westernmost population of the species, known as the Columbia white-tailed deer, once was widespread in the mixed forests along the Willamette and Cowlitz rivers valleys of western Oregon and southwestern Washington. But today its numbers have been considerably reduced, and it is classified as near threatened. The mule deer, Oedicolius hemionus, is a deer indigenous to western North America. It is named for its ears, which are large like those of the mule. Two subspecies of mule deer are grouped into the black-tailed deer group. Unlike the related white-tailed deer, mule deer are only found on the western Great Plains, the Rocky Mountains, the southwest United States, and on the west coast of North America, where they are best known as black-tailed deer subspecies. The most noticeable differences between the white-tailed and the mule deer are ear size, tail color, and antler configuration. In many cases, deer species body size is a key difference. The mule deer's tail is black-tipped, whereas the white-tailed deer's is not. These are all about appearances, not genome variations of alleles and DNA sequences. Mule deer antlers are bifurcated and they fork as they grow, rather than branching from a single main beam as is the case with white-taileds. We saw in discussions about elk, the Cervus canadensis variety nelsoni example, that elk were considered on the basis of a subspecies. In this topic of white-tailed deer, Oedicolius virginianus, and mule deer, Oedicolius hemionus, these species are in the same genus, but not the same species group. Deer of North America are more distantly related than elk. This has come as the description of Sympatric Speciation. It happened with the full resultant populations being geographically adjacent and sometimes overlapping. Throughout this speciation series, we have been looking at these chubby and happy Roosevelt elk, Cervus canadensis variety roosevelti, on the left side screen, opposing the Rocky Mountain elk, Cervus canadensis variety nelsoni. Members of the genus Cervus are descendants of Cervus genome first appearing in the fossil record 25 million years ago, during the Oligocene epoch in Eurasia. But they do not appear in North American fossil record until the early Miocene, about 23 million years ago. Think about the current ice age that started about 34 million years before present. Members of the North American Cervus genus have been developing within these restrictive environments since that time. The North American name bearer was Cervus canadensis variety canadensis, but it has been considered extinct since 1 September 1877, when the last elk bull was shot in Pennsylvania as a hunting season success. Oops.
Oh no! No! Retrieve the melon! Taekwondodos! Attack! The melon!
There goes our last female.
Subspecies of the Cervus genus are groups at the first stage of speciation. Individuals of different subspecies sometimes interbreed, but they normally produce sterile male offspring. At the second stage of incipient species, or semi-species, individuals of these groups rarely interbreed, and all their surviving male offspring are sterile. We see this incident with deer, and we already talked about. Roosevelt elk are the largest of the 4 surviving subspecies of elk, Cervus canadensis, in North America by body mass. Although by antler size, records have Rocky Mountain elk antlers being the largest. Think about that adaptation. Elk in the Olympic Peninsula live in temperate rainforests with thick understory plant communities, dense trees with branching to the ground. Big antlers may be advantageous to show intraspecies dominance, but when they get hung up in the understory while being pursued by predators, they may be a liability. This is an act of species selection to reward survivors. Rocky Mountain elk in Yellowstone National Park live in the temperate coniferous and hardwood forests and rangelands. Big antlers are not a liability to be hung up in the understory brush or on low-hanging tree branches. But they sure serve a purpose when being attacked by a pack of wolves. These 2 varieties of elk are in the first and second stage of speciation. They have demonstrated ample evidence of speciation adaptations to different environmental restrictions. I ask you now, what form of speciation explains this? Cervus canadensis variety roosevelti is genetically different from Cervus canadensis variety nelsoni. Let us look at each type of speciation. Allopatric speciation explained the process of separating Cervus canadensis variety nelsoni elk in North America from Manchurian wapiti, Cervus canadensis variety xanthopygus, in northeastern Asia. That happened when we left the apex of the current ice age to melt ice, glaciers, and lift global sea levels, a process that continues today. The geographic separation meant random genome mutations served to adapt to similar environmental constraints, but the solutions made different DNA alleles in the separated populations. In this Western North America example, there seems not to be a geographic separation forcing them apart. Elk live in the Cascades, Rockies, and rangelands throughout this continent. These are not barriers to separate elk. This one does not seem to fit. What about Peripatric Speciation? In that scenario, we look for one large population that is separated by something like global glaciation for super long periods of time, like centuries or longer. And then they are rejoined like the red spruce and black spruce were. Their genetic adaptations to restrictive environments demonstrated different adaptations to the rejoined ecosystems, which are now very similar. Seems that one does not fit too well here because the Olympic Peninsula and Yellowstone were not similar before and they are not similar now. If we back up the clock 100 million, 70 million, and 50 million years ago, we cannot even see the accretionary wedge coming off the Cascadia subduction zone to build up the western face of North American continent and the Olympic Peninsula. But I can see where Yellowstone was then and still is today. I think it is not Parapatric Speciation. The right conditions just do not seem to fit. So what about Parapatric Speciation? This scenario we explained with Darwin's finches. In Parapatric Speciation, 2 subpopulations of a species evolve reproductive isolation from one another while they partially continue to exchange genes. This mode of speciation has 3 distinguishing characteristics. 1. Mating occurs non-randomly. 2. Gene flow occurs unequally. And 3. Populations exist in either continuous or discontinuous geographic ranges. When we consider the geographic discontinuity from Forks, Washington, across the Cascade Mountains, through the Great Basin and into North Idaho, and then climb over the Rocky Mountains, and finally into Wyoming to find the Yellowstone hotspot, we see plenty of reason to recognize geographic discontinuities. There are still genes being shared between subpopulations, but it is not continuous, and success in one area will not perfectly translate into advantages in the next. This distribution pattern may be the result of unequal dispersal, incomplete geographical barriers, or divergent expressions of behavior, among other things. Parapatric Speciation predicts that hybrid zones will often exist at these junctions between the two populations. In biogeography, the terms Parapatric and Peripatry are often used to describe the relationship between organisms whose ranges do not significantly overlap, but are immediately adjacent to one another. They do not occur together except in a narrow contact zone. In this scenario, we would expect nearest neighbors to share genes and produce semi-viable offspring, but then to see unsuccessful offspring from participants at the far reaches of these subpopulations, like between an elk parent in Forks, Washington, mating with one from Roswell, New Mexico. Their genetic drift is expected to be significant for one another, and their offspring would not be expected to be viable, especially with males. Parapatric speciation is looking like a good option, but we have one more to consider: Sympatric. Sympatric speciation is the evolution of a new species from a surviving ancestral species, while both continue to inhabit the same geographic region. We consider this when looking at white-tailed deer and mule deer example. There was one species originally, and from white-tailed deer evolved the mule deer species, formed to fill the niche of higher elevations, steep slopes, feeding on more forbs and shrubs. Today, we see both species sharing space at Kamiak Butte, and throughout the Columbia River Basin of Washington and Idaho. Their habitats commingle in places, but species uniqueness is obvious. I do not see a commingling of Roosevelt elk and Rocky Mountain elk anywhere. Some of the subpopulations stream from western Washington and Oregon into the Cascade Mountains. Members are considered Rocky Mountain elk, but breeding intermixes with Roosevelt elk are possible. Roosevelt elk evolved within the temperate rainforest ecosystems, not in the high-elevation temperate forests of the Rocky Mountains. These ecosystems are sometimes nearest to neighbors, but they do not overlap. Seems the nearest neighbors may be able to reproduce, but the farther away the elk populations travel from these core areas, the less genetic expression resembles either core population genome. Sterile offspring would be expected as a result. I think we have a winner with the Parapatric speciation. It may have been explained by John Gould to describe Darwin's finches, but tape it to the elk in the Cervus canadensis genus, applying the familiar description of how populations find successes to survive and thrive. You may develop different ways to describe the speciation process, revealing various subspecies adaptations. When you do, use the define and discuss with example mantra to evoke the reasons for the choices made. Adaptive radiation is the process in which one species gives rise to multiple species that exploit different features of the environment, such as food resources or habitats. The different features of the environment exert the selective pressures that push the populations in various directions, and we call this phenotypic divergence. This leads to reproductive isolation, the necessary condition for speciation to occur. It is often a byproduct of the changes in morphology, behavior, or habitat preferences that are the actual targets of selection. Likewise, variations among local populations in biotic interactions can result in phenotypic divergence and therefore have the potential to function as mechanisms of adaptive radiation. Why could interbreeding be an important consideration? Well, think about genetic variation and how mutations are part of adaptations in specific environments. What if mule deer developed a mutation that is passed to its offspring, allowing it to better defend against predators? Or a black-tailed deer population mutated a heritable trait to efficiently digest noxious weed plants introduced to their environment? If that interbreeding transferred to a member of the other population, then new abilities to fill the niche would be possible. Where sexual reproduction is the only form of genetic transfer, the transference of genes with desirable heritable traits is paramount to survival. This plays out for all species, herbivores and carnivores alike. It also plays part for plants and fungi, encompassing all biotic life as we know it. Resources are often limited in a habitat, and many species may compete to get a hold of them. Elk along this river compete for food, nutrients, water, and space. They also compete for all these resources with deer, beavers, and other herbivores in the same space. We explored the concept of an ecological niche and saw how species having similar niches leads to competition. We explored how species can evolve by natural selection to occupy different niches, thus divvying up resources and minimizing direct competition.